Good morning, everybody. Welcome to day 3 on week 6 of the semester. Remember that this is the week of finals and that the final exam must be submitted by 11:59 on Saturday. So the summer semester for us ends on Saturday. So make sure that you submit all of the work due. I cannot give any extensions past Saturday just because the semester is over. So understand that, for example, if for any reason you forgot to do the labs yesterday, please send me an email. I'll be more than happy to help you, but I can only extend it during this week just because the semester ends on Saturday. Now, when it comes to material, the last thing that we mentioned yesterday in class was the idea of the central dogma of genetic information. When it comes to that, we introduced the idea that when it comes to nucleic acids, the genetic information is stored in the form of the nucleic acid DNA. And remember that DNA stands for deoxyribonucleic acid. Then, through the process of transcription, and as you can see in the notes, which happens in the nucleus of an animal cell and other organisms that have cellular components just like us, we are going to have that DNA be the template in order to make the other type of nucleic acid, which is called RNA. This process of transcription occurs in the nucleus of the cell. Now, once that RNA molecule has been made, it's going to undergo modification. So if you take upper level biology classes, you will see the different modifications that happen to the RNA molecule. You can see here in this particular figure that is labeled mRNA, because this is actually known as the messenger RNA because it is synthesized from DNA. And there's different mRNAs out there. Now, when it comes to this mRNA, remember that it's going to be transported from the nucleus to the cytoplasm. And in the cytoplasm, we're going to have different components of the animal cell. Specifically, we are going to have RNA components, we're going to have protein components that are going to come together through the process of translation. And in translation, what happens in the cytoplasm, then that mRNA is going to serve as our template in order to make protein, okay? Which is the sequence of amino acids that we know that come together in order to give rise to the different structural levels of proteins, which we studied already in the last chapter, which include primary, secondary, tertiary, and, if available, quaternary structures. So with that, let's now look in detail at RNA as a molecule. So in class yesterday, we learned that when it comes to DNA is really long. So just imagine our cells are packing DNA that if you stretch it out, it's about 6 feet long. But understand that when we compare length to quantity, DNA is actually not the most abundant nucleic acid in the cell. RNA, on the other hand, is what makes up most of the nucleic acid that we have in cells. Why is this other nucleic acid, RNA, ribonucleic acid, important? Because it transmits that genetic information that came from DNA in order for the cell to at times produce protein. Understand that, again, we don't talk about this in detail in this course, but for those of you that may be moving to other classes that are upper level biology classes, not all RNA is converted into protein, okay? There's some RNA that stays as RNA and is still utilized in the cell for cellular operation. Now, We already learned that when it comes to the basic unit for RNA is going to be a nucleotide, and this nucleotide is going to contain the ribose sugar that is phosphorylated at the 5' carbon. So the OH that is present at that position is phosphorylated, and that nucleotide is going to have any variation of the nitrogenous bases, adenine, cytosine, guanine, or uracil. Now, let's understand that when it comes to these nucleotides, they're going to be different from DNA. And we already seen some of these differences when we introduce the nucleotide structures of each of them, but let's just remind ourselves what are the difference between DNA molecules and RNA molecules. As you can see in item 1, remember that in an RNA, their nucleotides are going to have a ribose sugar instead of a deoxyribose. Then, when it comes to the nitrogenous bases, okay, which at times we just refer to them as bases. So in biochemistry, please be understanding for those of you that have to take other biochemistry classes that when people in their language, they say base, they are not referring specifically, if they're talking about nucleic acid, they're not referring to sodium hydroxide or potassium hydroxide. When they say a base, and we're talking about nucleic acids, we are referring to the nitrogenous bases. And as you can see in item 2, when we have a molecule of RNA, we do not have thymine in it. In its replacement, we have a nitrogenous base that is called uracil. So we're going to see a few moments through the process of transcription how the base pairing, whenever that RNA molecule has been synthesized, is going to be a little bit different than what we saw in DNA replication yesterday. Third, you can see that the RNA molecules, for the most part, are single-stranded. Okay, in DNA specifically yesterday, when we look at the secondary structure, we saw how most DNA is double-stranded. Again, this is just the basis of the information. As you guys progress in your studies, especially if you have to take more specialized courses in these subjects, you will see that there are also variations. So please don't be surprised if you take upper level biology that all of a sudden they say like, and there is this example of double-stranded RNA. There are special cases, they exist in nature. Or if you see single-stranded DNA, all of a sudden they'll be like, oh my God, Dr. Luz lied. No. Understand that these do exist. I'm just introducing you these concepts. So as you progress in your studies, you're like, oh, wow. So all this biology word is very, it's complex, what I'm trying to say. And there's more variation that, the initial information that we were studying. Lastly, RNA molecules in terms of length and size, they're going to be smaller than DNA molecules. So remember, as I mentioned a few moments ago, DNA molecules are really, really long and they're packed tightly inside of cells. When we look at how many bases we have in an RNA molecule, is going to be less than DNA. Now, it is important to note that when it comes to RNA, there are numerous types of RNA molecules, specifically in humans. But in this course, we are going to focus on these main 3 types of RNA molecules. Just to warn you guys, these RNA molecules do come from DNA, but these RNA molecules are not translated to proteins. They remain inside of the cells for humans as RNA molecules, starting with ribosomal RNA. When it comes to ribosomal RNA, as you can see, ribosomal RNA, which is the majority of the RNA that we have present in our cells, it is involved in the synthesis of proteins because it is a major component of this organelle, which is the proper word to define it. So this component of animal cells that is involved in the synthesis of proteins. So in the ribosome itself, you see these RNA molecules in it that are called ribosomal RNA. Now, we also have messenger RNA. So the mRNA that you saw in the previous slide, that's what messenger RNA is. So what is a messenger RNA? This is the RNA that carries information specifically for protein synthesis. And remember, it was synthesized through the process of transcription from DNA. All of this happened in the nucleus, but then that's the one that gets modified. And then there are several processes that it goes to, but ultimately it leads to the formation of proteins in the cell. So when you look at the percentage of that type of RNA that we have in human cells, it's about 5%. Lastly, we're going to see in a few moments how in the process of translation, that third type of RNA actually plays an important role. We have in cells what is called transfer RNA. And transfer RNA are going to be this single RNA molecules that fold in a particular way. It actually looks like a bobby pin, like a hairpin in the cell. And understand that their role is that during the process of making proteins, that type of RNA, tRNA, is what brings the amino acids to the ribosome to make that protein that has been synthesized using as a guide that messenger RNA that we have in item number 2. Now, we are going to look into detail. And as I mentioned to you guys, we are not going to look at the secondary structure of RNA molecules here in this course. I can just point out the secondary structures for some of them. But we are not going to study it in detail, just like we did DNA in yesterday's course. Now, what I want to proceed and talk about is the process by which we are making that ribonucleic acid from DNA. And as I expressed a few moments ago, DNA transcription, okay, which happens at the nucleus, is where specifically we're going to be making that RNA molecule, okay? So before, let me just show you guys a YouTube video that summarizes DNA transcription, and then we can talk little by little about all the steps that we're going to see in a video. So give me a moment to go to YouTube and just find an animation about DNA transcription specifically. So in the chat, I put the address for this DNA transcription animation video. So those of you that want to review the steps outside of the slides and want to see this animation, you are more than welcome to revisit the video. In addition, this is one of my favorite videos that I found on YouTube in recent years, just because it brings me a little bit of nostalgia. And those of you that have family or have grew up around novelas, you will know what I'm talking about when I play the music, because it has that novela factor. Like in the beginning of like any classical novela, you know, it could be Marimar or you know, the classic old novelas, you know, or like recently, like La Rosa de Guadalupe or something like that. It has that tone of like, you guys will see, and then maybe you will laugh. But let's watch the following video on DNA transcription. Wait, this is translation. Let me go back to they have a transcription one. Give me a few moments. I'm trying to find the DNA transcription one.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, Rna polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of Rna polymerase. Once Rna polymerase is bound, other transcription factors complete the mature transcription complex. Now energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
So I just shared on the chat the address for the transcriptional video, okay, just in case. Now, let's notice that the video that I just showed you guys, and you saw that there were different components of it, all of that was how transcription actually occurs in our body. So in animal cells, that's how it goes. The simplified version of the process of transcription that we have on the slides are specifically the transcriptional process that happens inside prokaryotic cells, meaning bacteria, because one of the things is that at the core of it, the steps are very similar. It's just that for us in larger organisms or in, organisms that have different components, or in other words, organelles in terms of their organization, understand that there may be a lot of factors that have to come together in order for the process to happen. But when it comes to understanding transcription, these are the steps that we need to be familiar with. As you can see, we are going to have, again, enzymes driving all of these processes. And the first enzyme that is very important in the system is going to be RNA polymerase. But in a way, it makes sense that we have RNA polymerase driving the process of transcription, because remember that at the end of the day, what we're making is a RNA molecule. We're making a ribonucleic acid specifically. So RNA is going to be the enzyme that actually brings together the, or that synthesizes, is what I'm trying to say, the RNA molecule. So this is an enzyme. Now, of course, RNA has to know where it needs to bind, okay? So you guys saw that there were different components. At least in our cells, we have enhancer regions. We actually have downstream, also regulators. We have a Tata box, But in general, understand that this RNA polymerase molecule is going to look at the DNA and know where to bind, because there is a sequence which is known as the promoter sequence, which is where RNA polymerase binds to identify what needs to be transcribed. Now, we learned yesterday in the process of DNA replication that we had a particular enzyme that actually unzipped the strands of DNA. Can anybody put in the chat, what is the name of the enzyme that takes or separates the strands of DNA during DNA replication? Does anybody remember the name of that enzyme? It's not RNA polymerase in DNA replication. What is the enzyme that separates the strands of DNA? Great job, you guys. Excellent. Helicase. So understand that RNA polymerase has actually helicase ability. So we don't need, at least in bacterial cells, a separate enzyme that is going to separate the strands. The RNA polymerase molecule is going to be able to do it. Now, you guys can see here how this is step 1. This is step 2. On step 3, now that the RNA polymerase has been able to separate the 2 strands, we're going to have RNA nucleotide aligned and basically use as a template the DNA. So when we focus on here, okay, I want you guys to notice that out of the 2 strands, our template strand for making RNA is this one. And the template strand is going to be the one that goes in the 3' to 5' direction. Okay, that is going to be the one that guides, okay, that RNA molecule that has been made. Now, for example, because in this image we only have the incorporation of 1 RNA nucleotide. If we have a adenosine molecule, because understand, especially for those of you that have to take other biology courses, adenine is the name of the nitrogen base. Adenosine is the name of the overall nucleotide, okay? So if we have an adenosine in our template strand, when it comes to specifically its base pair, meaning the strand that is being formed that is going to end up being the RNA molecule, this is going to base pair with uracil. Okay, so for specifically adenine, we're going to base pair it with uracil. Okay, so to give you guys a little bit of a example of how this will look like, let's say that this is U. Okay, if we have, for example, a cytidine or a cytosine nitrogen base in a molecule, that incoming blue piece that is going to be incorporated, what letter is it going to be? A, T, C, U, G, which one is going to be? So that blue piece that is incoming, that is going to be connected to that uracil. What nitrogen base is going to be in it? A does not base pair with C. Remember that your base pairing for transcription is, if you find an A is going to base pair with U. So if this is your DNA template, And then this is your RNA that is going to be synthesized. So if the DNA template has an A, then on the RNA, we're going to place a U. If we have a C, then this is going to be a G. If this is a G, this is going to be a C. If this is a T in the DNA, it's going to be an A. So if my template has a C, okay? Let me redraw this to be more clear, because this arrow is just showing that. So we already incorporated that T, okay? So if I have a new incoming nucleotide that is going to be linking with the U, but it's going to base pair with the C, what letter is it going to be? Excellent. I see where you guys got your U from, okay? That's why I erased the arrow, because I know where you guys were getting the U from. So if we already incorporate that U, the new incoming one, just like you guys put on the chat, is going to be a G. Okay? So let's focus basically, because in this base pairing, I just want to focus on what we have here. Let's say that this is a TAG, okay? So at the level of the RNA, what's going to be the 3 letters that are going to be present in the RNA if my template of DNA looks like a TAG? Great job. This is an A, U, C. One of the things that you guys need to know, and if you have taken other biology classes, you guys had to know how to do this. It's like if you're given the template, you need to give what is going to be the RNA and the protein and so on and so forth. If you guys have looked at the homework, you guys also have to do those type of exercises. And we're going to practice today on how to do that. So understand that when it comes to that molecule of RNA that is being synthesized, because the RNA polymerase is moving down the DNA to elongate that RNA molecule, we're going to use as a guide the letters in the template strand, which is the template strand. We are using the strand that is in the 3' to 5' direction, because remember that this molecule of RNA is being synthesized 5' to 3'. And we explained yesterday in class those chemistries that are actually explaining why is it synthesized in the 5' to 3'. For those of you that need a reminder of why is it that this thing is made 5' to 3', remember that when it comes to the functional groups that are available in order to be synthesizing, specifically this nucleic acid, we can see that the 3' hydroxyl group of the last nucleotide in my growing chain is going to bond to the phosphate. And as I mentioned yesterday in class, it is a triphosphate to the phosphate in the 5' of the incoming nucleotide. Okay, so that's why RNA, similar to when DNA has been synthesized, is made 5 to 3. Remember that all of that has chemical bases. Now, through the process of elongation, at some point, the RNA polymerase molecule needs to stop, okay? So we can see here in the elongation process, this is just another illustration of how is this elongating RNA molecule has been made. And as we just expressed, remember that that RNA molecule is going to be complementary, okay, to the template strand in your DNA. So how is it that the RNA molecules know where to stop? Well, there are different ways in which, depending on the organism, depending on the system that that RNA polymerase is acting on, there's going to be different ways in which the RNA polymerase stops the process of transcription. Here, I am just illustrating the use of the Rho protein to stop the process of transcription. So when a particular system that utilizes Rho as the way to stop transcription, what happens overall is that we have a protein inside of the cell that is called the Rho protein. Now, Rho is going to be binding to this nascent, meaning the one that I was just formed, RNA chain. Sorry, not the RNA polymerase, the RNA chain. So it's going to go in, okay, and is going to pull away that newly synthesized RNA strand from the RNA polymerase and the DNA template, okay? So the Rho protein knows, in its interaction with that RNA polymerase molecule is like, hey, you're done with your job. Thank you for making that RNA molecule, and the system basically comes apart, and then the RNA polymerase goes to whatever other segment of DNA that needs to go to in order to perform another transcription, okay? Any questions when it comes to the process of transcription? So understand that in this process of transcription, it happened inside of the cell. Once this RNA molecule is made in the nucleus specifically, chemically is actually going to undergo different transformations. We are not going to talk about those modifications, but basically at the 5' end, it's going to be modified. At the 3' end, it's going to be modified, because inside of the cell, we actually have enzymes, or that are protein based, that are going to start eating up that RNA molecule. So again, as I mentioned to you guys yesterday, biochemistry, biology, just science in general, is such an interesting system in the way that it was designed. So once that RNA molecule gets transported into the cytoplasm, it is going to undergo the process of translation in order to make protein. So understand that RNA translation specifically, or let me specify here that transcription is when we go from DNA to RNA. RNA translation is where we go from RNA to protein. And this specifically happens at the cytoplasm of animal cells. If we're talking about bacterial cells, transcription and translation actually happen in the same compartment, so it's even more interesting. Now, let me go back and find the video that I illustrated to you guys first, which is the one for translation, so we can actually see the process of translation in an animation. So give me a moment. And the link for this video is already in the chat. This is the first link that I gave you guys.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So as we just observed in the video, the process of translation has important key players in order for translation to happen. Now, the translation process that you guys saw in the video is going to be the one that is for animal cells. The one that we have here in the slides is going to be for simpler organisms like bacteria. But overall, it has very similar components. So to summarize the process of RNA translation that happens in the cytoplasm, let's understand that the key components that have to come together in order to synthesize those proteins are going to be having a tRNA molecule, okay, which this is one of the RNA types that we talked about a little bit earlier. And this is going to be the molecule that carries the amino acid. Let's understand that when it comes to the structure of a tRNA, the tRNA structure has what is called an anticodon or anticodon. The anticodon, as you can see, is what's going to base pair with the template, which is the mRNA. So specifically, understand that that anticodon is what's going to base pair with the nitrogenous bases that we have in our mRNA. So for example, if in your mRNA you have an AUG, the anticodon that exists in the tRNA is going to be UAC. Now, it is very interesting, again, if you take upper biology classes or biochemistry, these topics are actually discussed. In the body, we actually have a particular enzyme that its sole job is to make sure that the correct amino acid is placed on the tRNA. Now, what determines the identity of this amino acid is not going to be the anticodon. What determines the identity of that amino acid is going to be the codon that is going to base pair with. So understand that even though the anticodon is going to be guided by base pairing with the template DNA, the identity of that amino acid is going to be depending on the codon itself. And as we're going to see in a few moments when we study in detail the genetic code, we are going to see that AUG is specifically utilized for the amino acid methionine, okay? And particularly in many of the translational events that happen in our body, the first amino acid that is always placed, or I should say not all translation is actually mediated by the incorporation of methionine, but the majority is. We'll see that methionine is going to be the amino acid that we find in proteins in the beginning, or the very first one, I should say. Now, it is important to note that we need a whole machine. In this case, this is actually what is called one of the components that we observed in the animal cell when we first started talking about the structure of an animal cell. And we can see that the ribosome, which are these dots here, they're actually protein producing factories. Okay, so when we look at those little dots, in summary, what they actually are is these ribosomal units. So those that represent those two pieces, we have a large one and we have a small one. So in order for overall translating an RNA molecule to protein, we need mRNA, we need a tRNA, we need ribosomal subunits. As we observed a few moments ago, I mentioned to you guys that we have ribosomal RNA. And to be honest with you guys, even though it is not illustrated in this image, understand that the ribosomal RNA actually lives in those subunits. So let me just label it a moment. So this is the rRNA. It is actually inside of the ribosomal subunits in the ribosome. That's where we find that rRNA. Okay, so as we observed in the video, when it comes to RNA translation, we have an initiation, we have an elongation, we have a termination process. So in general, those steps are very similar to what we saw in transcription. Now, we observed that during the process of initiation, what we see is that specifically the components of the translational apparatus, which is those ribosomal subunits and that template RNA, are going to come together, and the RNA is sandwiched in between the two ribosomal subunits. Now, understand that at this point, once the ribosome has sandwiched the RNA molecule, then the tRNA brings in the first amino acid, which, as I mentioned, typically is the start codon, meaning AUG. In the process of elongation, the reason that tRNAs keeps on coming is that when we look at our molecule of RNA that is being translated, the ribosomal machinery actually reads it in threes. So it's going to look at these three, then these three, then these three. And that's how it's going to read overall the RNA molecule, okay? So as you can see here, once it starts incorporating those amino acids, that amidation reaction that we talked about in the last chapter, meaning the carboxy terminus and the amino from the amino acids, is what makes that amide bond between the amino acids. All of that happens at the ribosome. And understand that it is very fascinating for me as a chemist how the body can actually take functional groups that maybe in the laboratory are hard to combine, and it clicks them just right there and then, because the body utilizes catalysts like enzymes in order to allow these reactions to occur. So even though in the laboratory it is hard to make an amide bond by combining a carboxylic acid in an amino group, in cells, the ribosome actually does that over and over and over again. And we can see here that the amino acids are going to be incorporated one by one when the ribosome unit keeps on moving down the RNA molecule, reading it 3 nitrogenous bases at a time, okay? Now, how do we stop the process of translation? Well, during the process of termination, when your ribosomal unit encounters what is known as a stop codon, that stop codon actually is going to interact with a release factor. Understand that a release factor is not a tRNA. The release factor basically comes and tells the translational apparatus to come apart. And then, as you guys can see here, we have completed the polypeptide. In other words, the protein that needs, that was synthesized in the process. And then at this point, what we see illustrated is going to be the amino acids that are in it. So this is its primary structure. And inside of the cytoplasm, this is going to undergo all those other changes that we saw in the last chapter, that secondary structure, that tertiary structure. All of those are going to happen after the protein has been synthesized. And as you can see, because these are enzymatic processes and that ribosomal unit is an enzyme, understand that the translational components are going to be recycled. So those tRNAs that now don't have amino acids go back to the process of chemically being modified by enzymes to incorporate the corresponding amino acids so they can be reused, and the ribosome goes and moves to the next RNA that needs to be translated. Any questions when it comes to the process of translation? So let's now move on and talk about the genetic code, which is a topic that I just mentioned when I was talking about the process of translation. The codons which exist at the RNA, okay, they already have been determined for all 20 amino acids, okay? So understand that at the beginning of the RNA of the mRNA molecule, the codon AUG is the one that signals protein synthesis. And remember that this start of protein synthesis is if the system starts with an AUG. Again, if you take upper biology, you will see that not all systems start with methionine. Now, there are a total of 64 codons possible. And again, they are going to be a combination of A, G, C, and U. The reason why at the genetic code we don't see thymine, because remember that the template specifically for translation is going to be RNA, and there's no thymine in RNA, okay? We have 3 stop codons, UGA, UAA, and UAG, okay? And remember that codon specifically is what allows the release factor to come into protein and the translational machinery just to come apart. So when it comes to looking at the genetic code, this table illustrates what are going to be the combinations of the different nitrogenous bases that lead to the codons that encode certain amino acids. Let me establish that you guys need this table for the final. Please do not memorize it. I don't have it memorized. If I ever, whenever I was a researcher, needed to look into this information, honestly, I just whip out my little table and that's how I looked at this information. Pretty much the only one that I have memorized is just methionine and then my stop codons. All of the other ones I look up because as you can see, there are going to be variations, okay? Now, let's note that methionine, which we see right here, AUG, there's only one codon for methionine, which is AUG, okay? Only one. There is another amino acid that only has one codon and one codon only, and that is tryptophan. Tryptophan and methionine are the only amino acids that only have one codon. The rest of the amino acids, as you guys can see here, can have 2, 4, even 6 different combinations of codons that gives rise to those amino acids, okay? Like for example, if we focus on the left side of the table, look at how many codons actually are the ones that gives rise to the amino acid leucine. You can have UUA, UUG, CUU, CUC, all of those. As you can see, all the way to CUG, all of those can encode for a leucine, okay? So again, methionine and tryptophan are going to be the only amino acids that only have one codon for them. So among the other codons that we have here, we can observe how these are going to be my stop codons, and this is where the release factor binds. And then you see the wide array for the different codons that exist for the rest of the amino acids that we have in existence. Now, it is known that the genetic or the genetic code for amino acids, it is known as degenerate. What degenerate means is that there are multiple codons, and again, there's only 2 exceptions, methionine and tryptophan. But for the majority of the amino acids, we have multiple codons that will give us those amino acids. That's what we mean with degenerate, okay? At least in terms of biochemistry. So now that we have covered DNA replication, transcription, and translation, let's now actually cover all of those processes by taking the following example, okay? In the following example, we need to determine, okay, or complete the following statements that we have here. So we are given a single strand of DNA molecule, okay? That we are going to utilize as a template. Now, as you can see here, we're going to use base pairing rules to find the following sequences. The first one we're going to do is find the complementary strand for this sequence. We are going to be able, if we are given a single strand of DNA, determine what is going to be the RNA sequence of it. Lastly, we are going to determine what is the protein sequence of it, okay? So you guys will see that I will take these slides and make multiple copies of it, and that's how you will see it on the notes, because it's going to be a little bit on the crowded side as we progress answering all of these. So the first thing that we're going to learn is how to determine the complementary strand. So if we are given single-stranded DNA in the 3' to 5' direction, its complementary strand is going to be given specifically in the 5' to 3' direction, okay? Now, what is the base pairing rule when we're trying to do the complementary strand given a single-stranded DNA? Wherever you see an A, you will put a T, okay? Because basically this is just representing what's going to happen during DNA replication if we're focusing just on the leading strand. Every time we see a C, we are going to place a G and vice versa. So at this time, we are going to do this 3 nitrogenous bases at a time, because remember that in this single-stranded DNA that we have here, what we are given is just the letters that represents the nitrogenous bases. This does not include the sugars, but we know that there are deoxyribose linking them each nitrogenous base to one another via 3 to 5 phosphodiester bonds. So let's get started. What will be the first 3 nitrogenous bases that are going to represent that newly synthesized DNA molecule that is complementary to the one given? Great job. So this is going to be GGC. So we're going one at a time. What is the next 3? Great. A, T, G. What are the next 3? A, G, C. Excellent job. Let's finish the last 3. What is the next 3, you guys? Next 3. And the last 3. Excellent. Any questions? If we are given single-stranded DNA, can we determine the complementary strand of it? Put a 1 in the chat if you know how to do this. Great. Now, let's move on to the next step. In the next step, we are going to determine the RNA sequence based on our single-stranded template strand. Remember that the RNA sequence comes from looking at your double-stranded DNA, taking the strand that runs 3 to 5, okay, and we are going to do complements in that newly synthesized RNA molecule. The base pairing rules for determining the RNA sequence is going to be the following. Every time you see a T in the DNA, that is going to base pair with an A. Every time you see a C in the DNA, that is going to go to a G and vice versa. That's why you see the double-headed arrow. Now, every time you see an A, okay, in your DNA, that means that its base pair on the RNA molecule is going to be the U, okay? So taking that into consideration, remember that RNA is also synthesized in the 5' to 3' direction. Let's do the same exercise, 3 at a time. What will be the first 3 base pairs in my RNA molecule? Great job. This is going to be G, G, C. What are the next ones? A, U, G. Next one? A, G, C. What is the next one? Great. G, G, A. Next one. You guys got this. And last one. Great. Okay. Put a 2 in the chat if you are given a template of DNA that you can determine what is the RNA sequence. Love to see those 2s. Now, lastly, we need to determine what is the protein sequence, okay? Now, when we are determining the protein sequence, I think I was able to do all of this in the same slide. So great, I don't have to copy paste. Because since we are in online learning, you guys are not going to be drawing them out, and you're not going to be submitting structures to me. Typically, if we were face-to-face, you guys will be drawing out the peptide that is made. So when it comes to protein sequence, understand that you need to find the AUG at the RNA molecule, and you need to find the stop codon at your RNA molecule. Now, understand that for the purposes of the final exam, you may need to do this all the way through, meaning that I give you DNA and I ask you for the protein sequence, or I give you RNA and I ask you for the protein sequence. So understand that it could be any of those 2 ways to find that protein sequence. So as you can see here on number 2, we already determined the RNA sequence, okay, for our template DNA. So we are going to be looking specifically at number 2 for determining what is the protein sequence here, okay? So first, we're going to find the start codon, that AUG, because that's the start. Understand that start is not the code for the amino acid. AUG is going to be specifically methionine, okay? So when we look at our RNA, we notice that here is our methionine. So that is technically where the ribosome going to sandwich our little peptide in this case, because it is an example. And this is where translation is going to start. When we scan through the sequence that I gave you guys on number 2, we have a stop codon. For those students, like, how do I know where's the stop codon? You go to this table. As you can see, what we're looking is either at a UAA, UAG, UGA. When we look back at what we're working with, we have here the stop codon. So what this means is that if this little piece of RNA was about to be translated, let's put in the chat, how many amino acids will be in that peptide? 1, 2, 3, 4, or 6? Excellent job, only 3. Methionine, which we already determined that it is the first one, because we found the AUG. And then this amino acid, this amino acid. So where do I find the amino acids that are encoded by AGC? I go to this table. How I utilize this table is that I go first letter, second letter, third letter. Remember, we're looking for AGC. So I'm like, oh, the first letter is A, and then it's G. Oh, okay, so I'm in this corner of the molecule, and then, sorry, of the table. So when we look at this, AGC is all the way out here. Great job, you guys. So that second amino acid here, this is serine, S-E-R. Then we go to the next one, GGA. Go back to this table. When I have a GGA, then I go, okay, first letter is a G, so I'm down here. Then I'm all the way back here, because the second letter is a G, so I know I'm in this area. And if you look, GGA is glycine. Okay? So this peptide is composed of methionine, followed by serine, followed by glycine. It would be incorrect to put stop after the glycine, because remember that that stop codon does not encode for an amino acid. That is just the position where we have the release factor come during the process of translation in order to finish our translation. Now, the correct labeling of my protein sequence will be that this is the N terminus. This is the C terminus. Okay, so sometimes depending on the labeling that you may see on the final exam, you may see N, met, ser, gly, C, and then you see the prime thing, you may see that. Or simply, you just see the 3-letter codes or the 1-letter codes for the amino acid. And you have to remember in your head that the first amino acid that is given in my sequence is the one that is exposing is amino terminus. That's why we have the N terminus in it. The last amino acid in my amino acid sequence for a given peptide is going to be the one in the C terminus, meaning that its carboxylate group, it is being exposed. Put a 3 in the chat if obtaining the protein sequence given RNA or given single-stranded DNA is something that you understand clearly. Excellent. So this is an exercise that it is very important that you guys know how to do for the final. And tomorrow in the Kahoot, we will be reviewing. Now, this is not a perfect system. And even though the enzymes that are involved in these processes have proofreading ability, mistakes can happen. Some mistakes are really detrimental. Some mistakes are not as detrimental. So genetic mutations can occur. Now, many of the genetic mutations actually happen at the DNA level, meaning that sometimes, you know, it could happen, you guys. And then I don't think specifically that for maybe certain diseases or certain genetic conditions, we understand clearly how it happens, but for other ones, we really don't. But at times, if DNA polymerase and during replication makes an oopsie, meaning that is going to incorporate the wrong nitrogenous base during the process of replication DNA, that may result in genetic mutations. Now, genetic mutations that can be divided into two different mutations. We can have frame shift mutations. Now, frame shift mutations are always detrimental to the amino acid sequence, meaning that is going to affect the protein. Because in a frame shift mutation, which is what we have in the bottom, this is specifically when insertions or deletions to a nucleotide can happen. And as you can see here, we have the example of an insertion. And if you have a deletion, it's going to be a similar effect. Because now the way that DNA that forms an RNA that forms a protein is going to be read with a brand new way, okay, in that codon, then understand that this labeling of codon 1, codon 2, codon 3, codon 4 refers to the codon that will be produced. This is not saying that specifically this is the codon, okay? Now, understand that when it comes to insertions or deletions, we are going to have a shift in the way that our RNA molecule that was produced is going to be read in terms of codons, and that's why it changes the amino acid sequence. Now, we can have other type of mutations that are called substitution or point mutations. Now, point mutations are not always detrimental to protein structures because they may or may not affect the amino acids that are going to be encoded, okay? So as you can see here, a point mutation by definition is going to be the replacement of one nitrogen base from the template strand, okay, into the one that got replicated, right? Now, this is the thing. Let's understand that, let's say that you are making more DNA and all of a sudden at the DNA level, and I'm just going to focus on this to study the different types of point mutations. Let's say that at the DNA level, we have a TTC, okay? And then let's say that I'm interested into making more DNA and I let my DNA polymerase do its thing. But then when I sequence my DNA, which is something that when you're doing research, that's your bread and butter. That is something that you do every day whenever you are dealing with DNA and then your graduate studies. So I spent, like, out of the 7 years total that I was in grad school between master's and PhD, this was at least 6.75 years of my life looking at DNA, understanding, did my replication went well? Do I have the correct protein based on the sequence? And we have techniques to look at sequences of DNA. It's actually kind of fun because it's just looking at puzzles. It just becomes really sad when you find that there is a mutation and then you have to start all over. That's why it takes months to sometimes replicate the DNA if there's mutations happen. Anyways, so let's say that at the DNA level, the one that I'm looking at is TTC. I do my experiment, I get my results back, and when I look at the DNA sequence that I obtain after replication, I see that it is a TTT. Well, not all hope is lost, because what happens is that change of a C to a T results in making a codon at the mRNA level that encodes the same amino acid. So that is called a silent point mutation. So as you can see, the silent point mutation is going to be that substitution of the nitrogen base that results in the same amino acid of your original DNA. Now, we can also have what is called a nonsense point mutation. So what is a nonsense point mutation? Okay, similar. You have a DNA that you're interested in, you went ahead and made more of it, you get your results back, and then you notice that a particular base, in this case, we've had a replacement of a nitrogenous base, but at the protein level, it actually results as a stop codon. It depends on the type of research that you need to do. This may not be very useful. Okay, so a nonsense point mutation is going to be that replacement of a nitrogen base that results in a stop mutation. Let's move on to the last two types of point mutations, the missense point mutations, which they can be conservative or non-conservative. So when it comes to a missense point mutation, again, we are comparing these three letters to my original. So again, there's a replacement. A conservative missense point mutation after translation and transcription makes an amino acid that is from the same family of the original amino acid that you started with. This may or may not be useful depending on your type of research. So lysine is a basic amino acid. This point mutation resulted in another basic amino acid, arginine. On the other hand, if we have a non-conservative missense point mutation, again, we have a replacement of a nitrogenous base when we replicate, when we make more of that DNA, but after translation and after transcription, we see that now we have an amino acid that is from a different family. So when we compare this amino acid that is created by the replacement, we see that that amino acid, again, is from a different family. Threonine is polar, lysine is basic, so different families of amino acids. That is considered a non-conservative missense point mutation. Any questions about the different types of mutation? Point mutations in the 4 different classes, or frameshift mutations? The last topic that we're going to talk about in chapter 17 is genetic mutations in terms of what we can see physically happening to the organisms, okay? Anybody's with me? I love X-Men movies. Put a 1 in the chat if you love X-Men movies. Anybody? Anybody? Thank you, Francisco, for this. Thank you, Shelly, for the support. Thank you, Alexa. I love these movies. I find them super, super interesting how they, have made this world of these people are considered mutants because they have genetic mutations, but it actually gives them superpowers. Oh, there's a question in the chat about the body detecting and correcting coding. Actually, the enzymes that are involved in making the replication of DNA, in that case, DNA polymerase, or the enzymes that are involved in transcription, like RNA polymerase, have what is called proofreading abilities. They are not perfect, but they do their job really well. So we didn't discuss it in this class because you typically talk about those concepts in upper biology classes. But if there is an oopsie in the replication process, the body has the ability and it has the proofreading ability. So just imagine that these enzymes have like a spell check ability, and they can actually remove the incorrect ones. Sometimes it doesn't do it, just because the body at times is not a perfect system, but the body's ability has the ability to correct itself if it has done something wrong. But going back to genetic mutations, I wanted to bring up genetic mutations because at times we think about genetic mutations and the first thing that people think about, which is unfortunate, it is true, some genetic mutations can lead to diseases, okay? And there are documented diseases that are due to genetic mutations, and then depending on what happened to the nucleic acids, that can lead to to organisms, and it could be in humans too, that may not be viable, or if they are viable, then they are going to be suffering some consequences. Again, some are detrimental, but some are not. Now, I wanted to give you guys examples of genetic mutations. So I wanted to bring up just things that aesthetically we can actually identify and be like, oh, that's a genetic mutation, I didn't even know. So these are examples of genetic mutations that you may have observed in everyday life and not even realize it. So in humans, blue eyes, it is actually a genetic variation. So don't think that mutation is always negative. As you can see, having blue eyes is just a genetic variation between the two genes that you see here, HERC2 and OLCA2, and that's what gives rise to blue eyes. That's why, when generations happen, the blue eyes, sometimes you see it in a whole generation of families, is because it is already incorporated in the gene. Albinism, it is actually also generated by a genetic mutation. And you can see that albinism is usually passed from parents to child. And overall, the mutation, specifically at the level of the DNA, is going to lead to the disruption of the production of melanin. And that's why, and you can see here, this is a peacock that is albino, and there are examples at the zoo where you can see all kinds of albino animals. I remember I went to San Francisco to the National Academy of Science, really cool place. They have an albino alligator. It was very interesting to see. But as you can see, in albinism, the mutation disrupts the production of melanin, okay? Which is, as you can see, the pigment that protects the skin from UV rays. One that maybe you guys are familiar with, and no, your eyes are not deceiving you, is polydactyly. Put a 1 in the chat if you have ever heard or seen somebody with polydactyly. Oh, cool. Put a 2 in the chat if this is the first time that you see this. Your eyes are not deceiving you. Yes, this person has 6 fingers on each hand, okay? As you can see, polydactyly is actually a condition from which a person is born with extra fingers, and it can actually also be with extra toes, okay? And it has been found that polydactyly, where certain indigenous communities in Australia, in their families, you can see members that have polydactyly, both in hands and toes, or one or the other, okay? Now, we know that that when it comes to polydactyly, part of it is genetic mutation. Other times it has been related to environmental causes. In terms of the functionality of it, I don't know how functional is that last finger or toe is, or if it matters. Like, I don't know what happens if people decide to not have it anymore, if it loses its balance. I don't know that. I don't. Oh, there you go. Somebody said that upon removal, if it happens really young, that can be done. But again, as you can see, that is going to be a genetic mutation, okay? So with this, you guys, this concludes all of the topics for the final exam. So at this time, we are going to take a break.